Hello biologists, this is Lab 109, Modeling DNA, and let's get started. Remember, these are the big ideas for this unit. DNA makes DNA, that's what we're going to talk about today. DNA makes RNA, and RNA makes protein. Replication is what we're doing today, this process of DNA making more DNA, copying the DNA before the cell goes through mitosis. We want to think about where that replication is happening. You can see these are cells here. There are 3 cells in this photomicrograph, and the DNA is dyed blue. So you can see the DNA here is in the nucleus. So replication is going on in the nucleus. That's important to remember. Question 0 on your lab sheet is using the terms nitrogenous base, sugar, and phosphate backbone identify the structures of this DNA molecule. Now remember, we've been talking about the base pairs and how the base pairs line up. Those base pairs are thymine, cytosine, adenine, and guanine. The sugar is attached to the base here. It's a pentose sugar. Deoxyribose is a pentose sugar. And the phosphate group here, which links the backbone together, is right here in this kind of cartoony molecule. So now you can fill out the first part of your lab sheet right here under question 0. If your lab sheet doesn't look like this, you need to look in doc sharing. That's the gray bar in the upper right-hand corner. You should be looking at a lab sheet that looks like this. So for question 1, it says, in your own words, describe the base pairing rules of a DNA molecule. Write out the names of each of the nucleotide bases that bond with each other. I've written out the names of each of the bases here, and here's an excellent diagram that shows that adenine bonds with thymine, and there are 2 hydrogen bonds here, and guanine pairs with cytosine, and there are 3 bonds here. So make sure you write a complete sentence for this question explaining how these base pairs work. If you need help remembering, GCAT is the way we remember that G pairs with C and A pairs with T. Number 2 says, what does the word replicate mean? We can think of this word replicate in terms of its parts. The prefix re means to do again, or to do something over. If you're going to redo something, you redo it over. And dupla in the Greek or Latin root means two. So to make two again is what replicate means. So when we're talking about DNA replication, it means make 2 copies of the DNA molecule. Why do we need to make 2 copies? Well, as the cell is going to undergo mitosis, it's going to need an exact copy of all the DNA in the cell for the new cell that it's going to make. So we can see here the cell cycle in this diagram, and we can see in the S phase, the cell goes through DNA replication. And if you look at this pie chart here and kind of mentally cut it into 4 pieces, you can see that DNA replication takes up one of those 4 pieces. So you can calculate the percent from that. Why is it important that DNA replicates before the cell divides? Here's PMAT, the PMAT of mitosis here, and the last phase here is when the cell splits into 2. Now each of those 2 copies needs an entire set of DNA. So the cell has to copy all of the DNA, so each of the 2 new cells from the one old cell has an exact copy of all of the DNA that was in the old cell. So we're going to look at building a DNA molecule using the base pairing rules from this website, and you can pick up that website in the notes on YouTube. So here I am at the University of Utah Health Sciences site. I'm going to Molecules of Inheritance, and I'm going to build a DNA molecule. I'm going to start building, and it shows you kind of the top side view of a DNA molecule. I'm going to choose an A, and then I know that another A goes with T, and if I pick the wrong one, it tells me that. And I'm just going to keep building a molecule. You can do this on your own. And when you're done, you're going to get a message. Remember what that message says, because you're going to need it for another one of the lab problems. So have fun building your DNA molecule. Practice those base pairing rules. And keep going until you get a message. To answer number 4 on your lab sheet, you're going to need to read this paragraph right under here. Notice I just scrolled down from underneath the build a DNA molecule, and I'm right here. Here's your information that you need to answer question 5. Oops, that's the information for question 5. Here's your information for question 4. So read this paragraph, read this paragraph. So for question 4, you read your interesting paragraph on molecular machines copy DNA, and you found out how fast a strand of DNA replicates. For question 5, you found out how many strands of DNA would equal the width of one piece of hair. And this is the message that you got when you were building the DNA molecule. In real time, how many years would it take you, not your DNA molecules, not the machinery of your cells, how would it take you to build the molecule? You're much slower than your cells. If you missed this one, go back to the website here and build your DNA molecule. And you'll have to match a bunch of base pairs to get the message. So for question 7, A, T, G, C are complementary pairs. We're going to look a little bit closer at how these molecules stay together by going to this website and viewing a little bit of an animation. So at this point, you can pause this video, find this URL in the notes of YouTube, click on this, watch the video, and then you can answer question 7. Here's another view of the DNA molecule flattened, and you can see the bonds right here. So that's a hint for question 7. So for question 8, in DNA replication, a double-stranded DNA molecule is duplicated to produce 2 double-stranded DNA molecules. Which of the following is true with regard to these 2 double-stranded molecules? And you should know this one from watching the video. These are 3 postulated methods of DNA replication that there were in the beginning before people started experimenting and figuring out how DNA replicated. There's semi-conservative, which means half is kept. Semi means half, and conserve means to keep. So half is kept. There's conservative, or there's dispersive. So if we go down and look at your multiple choice options, you can probably guess which one is correct based on what you just saw in the little tutorial video. I can demonstrate a little bit with a zipper here. If we consider the zipper here to be the DNA molecule that we want to replicate, we consider this fabric part, the backbone, the sugar phosphate backbone of the DNA, and the teeth, the base pairs. A helicase molecule will come in and basically unzip the molecule. And then DNA polymerase will work with half of the old DNA, the DNA we want to copy, and start bringing in base pairs one at a time and replicating. So this new DNA molecule is half of the original DNA molecule and half new base pairs. So let's go look at the animation of replication one more time. We've seen this before, but it's important now to look at the role of the enzymes. There's a whole cast of characters here that are enzymes in replication. So pay attention to what's going on down here. These are a cast of characters of enzymes that are important in making replication happen. So let's watch what happens first here. This green rectangle, which represents helicase, comes in and unzips the DNA molecule. It breaks the hydrogen bonds between the base pairs. And then polymerases start building the DNA molecule. And you can see that it's going to happen in two directions, depending on which side of the DNA molecule it's happening. This side goes this way, and this side heads in this direction. And you can see all those enzymes come into play again. I'll reset it here. Go back to the beginning so you can see all of the enzymes. You should play this on your own. I'll put it in the notes for the YouTube so you can go in and look at that replication video one more time and look at the role that these enzymes play. So you might want to pay special attention to the green enzyme and the blue enzyme here when you go and look at that replication video on your own. Those are especially important enzymes. So for question 10, we're going to talk about how DNA replication is like a zipper and not like a zipper. Well, it's like a zipper because DNA kind of gets unzipped. It has two sides. It has teeth that stick to each other. And it has something that breaks bonds and makes bonds. This pull here on the zipper would be an enzyme. The backbone here would be the fabric where you attach it to your garment. This would be the sugar phosphate backbone. And these are the bases here. The teeth of the zipper would be the bases. Now there are some weaknesses to the model. Obviously, there's different enzymes that unzip and zip the DNA. So our zipper bowl has to play a couple roles here. And when we start replicating the DNA, we don't just bring in a whole new piece of DNA. Polymerase would bring in one base at a time, one zipper tooth. But it is a good model, especially when you have two colored zippers here, because we could see semi-conservative replication. We can see that half, semi, is the old DNA molecule that we're copying, and half is new base pairs. So hopefully you should be able to write at least two complete sentences for number 10 at this point. Number 11 asks you what semi-conservative replication means. Here's a picture, and we know that semi means half, and conserve means keep. So we know that semi-conservative replication means keeping half, but it's your job to explain what that means in terms of DNA. So the optional bonus question asks you, what happens if there is a mistake in replication? And here's a really easy way to find out. www.nature.com, and I'll put this URL, of course, in the notes as well. You can read all about how DNA replication and causes of mutation and what cells do to correct mistakes made during DNA replication and how they work and what happens if those systems fail. So that's a great bonus question. I believe it's actually worth 2 points. And there's also another bonus question on what kind of molecule is an enzyme, and you can look that up too. There are some different options on your sheet. So thanks everybody for coming, make sure when you finish your lab sheet, you put it in the drop box. You picked up your lab from